Okay, so welcome everyone, and thank you for joining us today for this webinar, where we will be talking about how to enhance our cell immunolabeling experiments using Fluigent's product, the automated sequential injection system, also known as Aria. So joining me today will be Maxime Poinceau, who is a PhD student also at Fluigent, and myself, I'm Amar Tamra, and I'm an application engineer at Fluigent. So first of all, let me introduce you to Fluigent. Who are we? We are a French company based in Paris. We develop and manufacture here at home microfluidic products that are destined to be used in applications where we need fluid control. We also sell these products, of course, and we support after sale our client and help them to develop their specific needs and for specific applications. So we are a world leader in the microfluidics market, and we are very well known in France and in Europe, but we are also looking to expand in the United States and in Asia. So we have also distributors, over 10 distributors in Asia and Australia. Our company was founded in 2005, so we have more than 15 years of field experience in the domain of microfluidics. And if you go ahead, for example, on Google Scholar and you look for papers mentioning Fluigent products, you will find more than 2,500 papers in journals such as Nature, Cell, and Science Advance. So we offer our clients unlimited potential applications depending on their needs. So this is the outline of today's presentation. Of course, I will start with a brief introduction about Fluigent expertise in the field of microfluidics and organ on a chip. And then I will introduce you to the topic of our webinar today, Aria system, which is an automated sequential injection system. And Maxime will be sharing with us his success story using Aria for immunolabeling of neuron cells. And at the end, we will have an interactive question and answer session. So if you have any question during the presentation, please do not hesitate to send us via the Q&A panel, so for us to answer it at the end of the session. So let's first start with how microfluidics is reshaping biology. If you want to define microfluidics, like Whiteside said in 2016, defined it in 2016, it's the science and technology of systems that process and manipulate small amount of fluids or liquid using channels with very small dimensions, tens to hundreds of micrometers. So this microfluidic, whether it's used with 2D models or 3D models, including spheroids, organoids, extracellular matrix, for example, is revolutionizing various applications, especially in the biological field, whether it's tumor immunology, tumor cancer studies, stem cell differentiation, immunology, and so on. One of the fields that microfluidics has been revolutionizing is the organ on a chip technology. So as you may know, many researchers use the 2D cell culture, which is a very robust model, but has proven to be not physiologically relevant enough to show what's happening in real life in the human body. So researchers have used also animal models, which are much more complex and relevant to what's happening in vivo, but still they are ethically debatable. And a lot of, for example, drugs that tested positive using animal models are not as relevant for human, for example. So recent studies have been developing other kind of models to be used for biological applications, such as 3D cell culture. And of course, once we incorporate microfluidics to this system, we will be talking about the so-called organ on a chip. So organ on a chip technology is a preclinical model for biomedical investigation, which will simulate what's happening in vivo. It will allow us to simulate the physiology, pathology for in vitro fundamental research, disease modeling, and drug screening. So it is really a revolution for the drug development pipeline. It allows early drug discovery, preclinical screening, and translation of new drugs. And it will allow the bridging of the gap between animal models, animal studies, and clinical trial involving human subjects. And of course, as any new technology, we need to continue the effort in integrating novel technologies for this model, including, for example, automation of the protocols, long-term liquid handling, integration of sensors, and using patient-specific sets. So like I said, one major component of organ on chip is microfluidics. So what are the advantages of this microfluidic 2 organ on a chip? Of course, it's the flex. For example, we have the flexibility of the device design. Many research are using their own device, their own design based on their application needs, which gives us a wide range of different design that could be used. It allows also the single cell analysis. Of course, due to the heterogeneity between different cells, researchers are more focusing on the single cell analysis in order to extract conclusive data. Excuse me. It allows also real-time analysis. So we can imagine the experiment happening under some kind of microscope in the presence of microfluidics, and we could see, especially in the case of long-term cell culture, for example, under the microscope, and we could extract significant results. It allows also the automation and the easy coupling to downstream analysis system. And of course, it allows perfusion culture and analysis. And it comes along with some kind of challenges, of course, like any new technology. So like I said, because we use different kind of devices and different kinds of protocol, so we don't have really these standardized protocols used by every researcher. So this is one of the challenges that microfluidics for organ on chip face. Another challenge is the use of novel culture surfaces. So some researchers use PDMS, others use glass silicone, others use thermoplastics. So we have a great variability between different tests done. And of course, add to that, it could be time consuming and economically consuming as well. So if we come and add living cells also to our system, we have to consider many risk factors, especially, for example, the cell type. Some cell types are more vulnerable than others, more sensitive than others, like, for example, of neuron cells. We have to take into account the surface properties of the device we are using and the various protocol steps that we are using, and to take into the account, of course, the shear stress and the flow rate control applied to our cells. So researchers have been using now different kind of fluid injection system. Of course, we have the gravitational rockers, which are very easy to set up, easy to scale up, but we don't have any control over the flow rate, and it doesn't allow any long-term experiment. Syringe pumps are also widely used because they are very easy to set up. However, the flow rate control is very low. We are very limited in terms of volume, and it will not allow long-term experiments. Peristaltic pumps are also widely used because they are easy to set up. It allows injection of large volume of fluid, but it is very low reproducible, and the flow rate control is very poor. This is where we come as Fluigent, that we procure pressure-based flow controllers, because these pressure-based flow controllers will allow controlled perfusion for biological application. they will allow a very stable flow rate and fast response time, which will give us also the ability to have a fresh supply of nutrients and oxygen over time, and will allow, of course, the long-term experiments from few hours to few days and even to few weeks, especially for organ-on-a-chip applications. So what is the technology behind our pressure-based controllers? It is a pneumatic technology, so we don't use any mechanical part within the system. So as you can see here, we have our pressure source, or we have our pressure pump here, which will be connected to a power supply, and it will be connected to a pressure supply. So this pressure source will supply the pressure for the system. It could be, of course, the pressure generator, gas cylinder, compressed air. The pressure here will be regulated by our pressure pump and injected inside a very well sealed reservoir containing the liquid of your choice, for example, cell culture media. And this pressure will help move the liquid inside the tubings, inside the microfluidic setup, and it will go through a flow unit, which is a flow sensor, which will allow to monitor the flow rate in the channel and communicate in real time with the pressure pump in order to adjust the pressure or the flow rate that we need. And then the liquid will go through the microfluidic chip where we have, for example, our cells or our organ on a chip. So the advantages of the pressure-based flow controllers is, of course, a very high stability. So if we look at the flow rate over time, and we compare the result we have with Fluigent in blue, Fluigent product in blue, and the high precision syringe pump in orange, you can see that the flow rate over time is very, very stable, very high stability. And also, we have a very fast response time over time. So here with Fluigent pressure controllers, it's it's a little less than 30 milliseconds, the response time. However, with the syringe pump, due to the inertia present in the pump, the response time will be up to few minutes, even a little more, to be able to stabilize the flow rate. And of course, when we use our pressure-based controllers, we can create very complex fluidic models based on our applications needs. So in Fluigent, we propose different solutions for different applications. For example, for cell culture and organ on a chip, for droplet and particle generation, whether it's single emulsion or double emulsion, for microfluidics in general, and for omics, which includes the tissue imaging, for example. So for today's webinar, it goes under these two categories, organ on a chip and tissue imaging, cell imaging. So I will leave you with this video that will explain a little bit about Aria, the automated sequential injection system, how it works, and then we'll take it from there.
Introducing Aria, our perfusion automation tool. Aria makes it possible to automate delivery of up to 10 different solutions in a petri dish or microfluidic chip by following user-defined protocols. Aria's precision makes it possible to inject defined volumes at specific times and to provide time-based sequential perfusion of multiple fluids under controlled flow conditions. Aria's intelligent software allows you to design the protocol that best fits your experimental model. Reducing manual sample handling and procedural complexity is key to improving precision, increasing reliability, and saving time. Even the cleaning is automated. Aria perfuses successive pulseless flows of different solutions, avoiding the extensive mechanical stress associated with classical pipetting. Aria automates protocols such as immunostaining by programming the time delivery of primary and secondary antibodies, the injection volume and flow rate, as well as the incubation time. Aria is the versatile ally you never had.
So at Fluigent, one of our main missions is to provide user-friendly and very easy to use and accessible microfluidic product, especially for biological applications. We know that biological experiments can take a lot of time, a lot of effort from the researcher's day. So our main objective with this kind of product was to we take care of the microfluidic part, and then you can focus more on your biological application. So really to make microfluidics very accessible to biologists especially. So like they said in the video, with Aria, we have a very high precision because we can, we have a full control over the fluid injection parameters. So for example, we have control of the flow rate, of the volume to be injected, and the timing, at what exact time we want to inject our fluid, our solution. It is very user-friendly. So it could be manipulated either manually via the interface here, or it could be also controlled via the software of Aria. So it doesn't need any kind of expertise or training required. It's very user-friendly, easy to use. It is versatile because we can inject up to 10 different solutions. So here we have 8, for example, and then here we have 2. We can inject up to 10 in one microfluidic channel, or if we add one rotary valve here, I will show you later, we can inject up to 9 microfluidic channels. The results obtained with Aria are very reproducible because we have a negligible variability between experiments, and of course, like we said, it is very time-saving device, and also reagent saving. Yes, so like I mentioned, we have 2 versions of the setup. We can connect it to one, the 2 switch, which is a valve that will allow the liquid to arrive here and then to go into the one microfluidic channel. Or if we connect it to the M switch, which is the rotary valve, and we have 9 outlet, excuse me, and these 9 outlets would go into 9 different microfluidic channels. One feature of Aria, which is very interesting also, is that we can synchronize it with any kind of microscope if we use TTL signals. So we would just have to connect Aria to our microscope, and then it could be like, it could say, you can program your protocol in a way that you would say, I want to do this cycle of imaging, and then followed by another cycle of perfusion, for example. So really, the applications with Aria are limitless because any protocol that you need to have a perfusion of some kind of fluid could be used. So for example, immunolabeling, long-term cell perfusion, DNA hybridization, the fish hybridization, or in vitro drug screening. So these are the numbers to keep in mind regarding the product. We have 3 main components: the pressure controller, the rotary valve, and the flow unit, in which we will monitor the flow rate that is being injected. The minimum volume required is 40 microliter, and the flow rate range is from 1 microliter per minute to 1 milliliter per minute, depending on the flow unit that we use. The maximum pressure is 2 bars, and we can inject up to 10 different solutions in one channel with the 2 switch, or 9 channels with the M switch. So now I will leave my colleague Maxime. He will be presenting for you his application, which is the brain-on-a-chip automated immunolabeling. So he will be using neuron cells for his application, and he will let you know more details about his application. Maxime, the floor is yours.
Thank you, Amar. So like she said, we use brain-on-a-chip to study the axonal plasticity. So just very quickly, what is a brain-on-a-chip? It's, you know, a PDMS chip used to mimic one or several aspects of the brain. So it's only in vitro culture with compartmentalization of the soma, the axon, and the dendrites. So that's it. We can continue. Thank you. What do we use? Why do we use brain-on-chip on the lab? It's because the big project of the lab is to have we study different molecular constructs to manipulate the cytoskeleton of neurons on demand, and the great goal is to be able to in adult brains to allow the regrowth of neurons after tragic event or neurodegenerative disease. So there is a video here where we can see an example where we have transfected our neurons with a molecular construct, and you can see that axons are growing everywhere. The culture is pretty old, more than 21 days in vitro, and the acquisition is realized within the time range of 20 hours with 5 minutes frame between the images. Thank you. So we needed a new brain chip specially designed to study the axonal plasticity. So the hypothesis was to take the structural formation of the corticostriatal network, where you can have two different cortical regions projecting their axon inside the same striatum, but two different and distinct regions of the striatum. So we created these chips where we have one cortex projecting one striatum and one other cortex projecting the other striatum. So this is to study the evolution of the plasticity and the evolution of the architecture of the axon inside the striatal chamber. So what does it mean? It means like here we have cortex transfected with GFP, and we can see a picture of the striatal chamber where we can see all the axon from the first cortex on the left reaching the left side on the chamber, the axons from the right side reaching the right side of the striatal chamber, and there is a big gap in between where there is no axons of the cortex, but there is striatal cells everywhere. And we want to manipulate the architecture to get rid of this gap and to connect or disconnect cortexes from their regions. So this is an image done after immunolabeling, but immunolabeling in microfluidic is very long because you have to be very steady and to control the flow rate. But with pipetting is very complicated, so it takes up to 5 hours because I need to inject a little bit of fluid, wait for the flow to stabilize, then take out some fluid, then add another little bit of fluid, to have a good immunolabeling. So I don't have any control of my flow rates. And we needed an automated protocol because we have tons of microfluidics to immunolabel and a lot of molecular construct to test. So yeah, like I said, time consuming, the uneven fluid delivery, pipetting errors if I just add too much fluid and then it's overflow everywhere, and solution mismatch. And the advantages of Aria, like we see in this picture, is the time saver, steady fluid delivery, sequential distribution in every chamber, and high reproducibility. So this is an example of manual immunolabeling if I don't take enough care. Before injecting the PFA sucrose, the cells are living their best lives. And if I had the PFA sucrose too quickly with my pipette, I destroy all the cells and all the axons, and the axons are very fragile. So this is a setup we came with the Aria software connected to Aria. On Aria, you can put the permeabilization solution, the blocking solution, and all your different antibodies, up to 5 antibodies. This is connected to the M switch, where the M switch is connected to the chips. Here, I only represented one chip, but with the 9 output, I can perform the immunolabeling of 3 different chips simultaneously. So it's a huge time saver. And then I go to the fluorescent microscope to then image the cells that I immunolabeled. So this is my setup at the lab. You can see the computer with the protocol, the Aria with all the solutions, and the M switch just at the entrance of the microscope. And I put the cells in the microscope for them to be in the dark not to bleach all the antibodies. So this is a protocol. We can have a quick look with the first type of permeabilization, injection of the permeabilization solution, then the waiting for the permeabilization to act. Then we have a loop here. The loop is kind of important because you really want to wash all the permeabilization solution. So I wash it with PBS. Then I had the blocking. I wait for the blocking. Then I wash the blocking. And then I had my antibody, my first antibodies, I wait, and the second antibodies, and so on and so on. So this is some results using automatic immunolabeling. So on the left side, you can see cells with anti-map to see the cytoskeleton of the cells, and Hutch to see the nuclei. And on the right, the big pictures is an artistic image of the axons of the cortex invading all the striatal chamber with a particular molecular construct that allows the cortex to grow very, very, very far from the cells after 21 days in vitro. So this is working very great. Since the immunolabeling is working very great, we have strong results on the structural regionalization of our experiments. But we want to do some functional experiments, and the question is, okay, I can grow cortex, but does my cortex create functional connection with the striatum? So the striatum is very silent in culture. It does not pulse like cortex without electrical activity. So here I have a video of a striatum that is pulsing. It's because cortex is directly connected to the striatum, and the cortex is giving input to the striatum that allow the electrical activation of the striatum. Or maybe you go too quickly and you pass the slide.
1 second.
Yes. One before again. Okay, this one.
Here, okay.
Yes. As you can see, here is a fluorescent probe that allow to see indirectly the electrical activities by the flowing of the calcium input. So every time there is a flow of calcium, the fluorescent probe is activated. So the striatum is very well activated. Then I add TTX. So TTX stop all the activity of the cortex. So I put TTX only in the cortex, but at the striatum is silent. Without any input from the cortex, the striatum is silenced. And then I add TTX in the other cortex to see if there is parasite connection, or if I don't want any connection. And you have to imagine that for now, I have my microfluidics inside the microscope, and I'm going with the pipette, just putting slowly the TTX in the cortex without moving to avoid losing my acquisition frame. So it's very complicated, and we want to use Aria to automate this process of neuronal activity. So let's look at this video to just have a glimpse, to give you more information about what I said, just to see the process of Aria. So we start the injection delivery. So we can imagine that the blue solution is the permeabilization solution. So it's flow through your cells like that. And then you wait for the permeabilization to take act. Then the red solution is the PBS, washing all the permeabilization solution. Then the yellow is your blocking. And after that, you can have your antibodies. So the take-home message that we really have good results with Aria. I don't lose chips anymore. And when we have an internship at the lab, I don't have to take a lot of time to form the intern to do this tricky immunolabeling. I just have to show them the protocol on Aria. It's very time efficient because I can launch 3 to 4 Aria protocol in a day. So I can immunolabel up to 16 microchips in a day. So it's very great, and I can let them run all the night, and when I come back on the morning, my cells are ready to be imaged. And it's game changer because I have all the time in the world to do other experiments while my cells are immunolabeled. So thank you for your attention, and we are here to answer all your questions.
While I would just like to draw your attention that on our website, we have various webinars that we already talked about Aria with different kind of applications. So if you are interested, you can go ahead and watch the webinars also. And we have some very interesting application notes also done with Aria. And most recently, we have published the application note of Maxim's work. So if you want to have a more detailed look at his work on his protocol or on his results, you can also find the application note on our website. I don't think we have any questions. Anyway, also, we will be sending you emails with access to our calendars. So if you want to have a more personalized discussion, one-on-one discussion with me or any sales representative, you can go ahead and book a session on Calendly. And we would be more than happy to discuss and to see how we can build your setup for your application. So I think that would be it. Thank you for your presence. Thank you, Maxime.
Thank you.